Welcome to Life and Biology. I'm Dr. Joel Graff, and this episode we're going to be talking about genetic linkage, how things look a little bit differently if genes are on the same chromosome versus on different chromosomes when we're doing things like Punnett squares or whatnot. So anyway, let's take a look at this picture. Let's just focus on the first half of the picture right now. And so we've got a cell that has a couple pair of chromosomes. We've got a shorter pair and then a longer pair up here. On the shorter pair of chromosomes, you've got a gene on it called A, and you can either have a capital A allele or a lowercase a allele. On the second chromosome that we're talking about, we're going to be following along with what happens with two genes on this chromosome. One is called B and the other one's called D. I skipped C because you can sometimes be difficult to tell a lowercase c versus an uppercase C. But again, we've got uppercase B and lowercase b, uppercase D and lowercase d alleles on that gene, or in that cell. If this is meiosis 1 and you've got your centromere over here, and you pull those chromosomes apart, we will end up with cells where you've got an orange and pink chromosome. That was what was on this side of the cell. And then we've got a blue and a green chromosome, and that ended up in this daughter cell. So that's what it looks like at the end of meiosis 1. And then, of course, you go through meiosis 2. It looks a lot like mitosis, and these X get broken apart so that you don't have a copy of each chromosome when you get your daughter cells. But I've left that out here. But bringing in the other picture, we've got the same sorts of chromosomes as in this cell. It's just that in this cell we've got orange on the left and blue on the right. This cell, orange was on the left and blue was on the right. And then the second chromosome in this cell, we've got green on the left and pink on the right. And here we flipped it around. We did pink on the left and green on the right. And so if you remember back to our laws, Mendelian laws, the ability for those sister chromatids or those homologous chromosomes to line up independently for this set versus that cell set, that was the law of independent assortment. So I've got 3 genes here to talk through. And we've got gene A is on this chromosome, while genes B and D are on this second homologous pair of chromosomes. And what we want to do is we want to think about what would happen to genes A and B if we did a cross between some cells that had this sort of chromosomes versus what would happen to genes B and D. And those obviously are linked together. So we're talking about genetic linkage. Those are linked together because they're on the same chromosome. So let's start off talking about gene A and gene B. Now, if you were to set up a dihybrid cross, this is the infamous dihybrid cross, where we've got gene A and gene B. We're doing a cross between 2 parents. The first parent at gene A had a capital A and a lowercase a, and at the second gene had a capital B and a lowercase b. Second parent also hybrid, heterozygous, at both the A and the B genes. So they've got the heterozygous alleles, okay? So when you go to make some gametes out of this cell and this cell, one of the A's will end up going, each gamete will end up getting one of the types of A's, and each gamete will end up getting one of the types of B's. And so we'll start off by taking the first capital A and this first capital B, and we say we've got a gamete that has that combination. We can then do, this is almost like FOIL, if you remember back to math class. We take the first A and the last B, the outer and outer, and we end up with capital A and lowercase b. Now we got to do our inner and inner, so we can take that lowercase a and the capital B, and that could end up being in a gamete. And then finally, last times last, you have this lowercase a and this lowercase b could end up being passed on in a gamete. And so we've first time first, outer, outer, inner, inner, last, last. And then that is the 4 types of possible gamete combinations you can have for genes A and B from this parent. Since this parent has the same genotype as this parent at both of those genes, then if we go down this side, we've got capital A, lowercase b, lowercase a, capital B, and lowercase a, lowercase b. So go ahead and pause the video if you need to write those down. And then when we take this gamete and we fertilize it with this gamete, we end up with 2 capital A's and 2 capital B's. So we're back to a diploid organism that is homozygous for capital A and homozygous for capital B, those genes. And then we can take this capital A and that capital A, mix it with this capital B and that lowercase b, and that's what we end up there. And I'm not going to walk through it, but if you want to just try to fill one of these out on your own, also they're littered all over the internet. This dihybrid cross is all you need to do a Google search for. But anyway, you end up with a 4x4 grid. You've got 16 different squares here. Some of the squares are the same, like this square and this square have lowercase a, uppercase b, lowercase b. And so they're the same. This is the only one that looks like that. And anyway, so you've got those 16 possible squares that we walk through. And in this case, if you're looking at phenotypes, the dominant trait for A and the dominant trait for B would happen 9 out of the 16 times. So if we went through and we found all the ones where there's at least a capital A and a capital B in the square, there would be 9 of those. There would be 3 that had the dominant A but both lowercase b, and there'd be 3 that had some sort of dominant B and then both lowercase a. So there's a 9 to 3 to 3 to 1 ratio for phenotype at least. And if you remember back to our monohybrid cross, there was a 3 to 1 ratio. So these ratios, because these are independently sorting genes, everything has an equal chance of happening. You're going to have that 9 to 3 to 3 to 1 ratio for phenotype. But that is assuming that those 2 genes are acting independently of each other. And so just to bring it back to our picture, A was on this chromosome, gene B was on the second pair of chromosomes, and so those are able to sort independently. Now let's switch our thinking to the genes B and D. They're on the same chromosome, so they're genetically linked, and normally with what we've talked about so far, you would think that those would always travel together. But that brings up the idea of genetic linkage. And one thing that we kind of skimmed over, haven't mentioned yet with the meiosis, is that when your homologous chromosomes, you have your 2 sister chromatids next to each other, they sometimes form what's called a chiasma. And that's where a similar sequence on the 2 genes kind of match up to such an extent that when these homologous chromosomes get pulled into opposite cells, they swap the tail bits. And so we've got a pink chromosome that ends with green and a green chromosome that ends with pink. And they swapped that last little tail of chromosome that was past that chiasma point. And so we're on these drawings, I'm leaving out the chromosome that had chromosome A, or the chromosome that had gene A. We're just focusing in on that one chromosome, what's happening with that one, because it's complex enough as it is. We're focusing in on just that one chromosome. So going back to this figure, we're ignoring the whole cell part, we're ignoring the green and the blue chromosomes, we're just focusing in on the green and the pink chromosomes from this picture. Okay, so I've given a couple different examples of what could happen. Up here, I've drawn it so that where the pink and the green chromosomes cross, they don't cross by much. And then in this picture, I have it drawn so that the pink and the green chromosomes, the chiasma is further up in towards the middle of the chromosome. And the thing about that is we're following these genes B and D, and they are both above the point where the chiasma was here. The two lowercase d's, for example, here from the pink one, they're above that chiasma crossing point, the crossover point, and so they stick together. And so the capital D's stick together. So the crossing over didn't affect how the B and the D gene were getting sorted. In this other example that I drew here, we do have the chiasma happening above where those two D genes occur, so that then we end up with kind of an odd man out where this capital D is on this chromosome where everything else is lowercase, and then this lowercase d is on this sister chromatid where all the other genes are the capital version. Hope this is coming across clearly in the video. But then this is what it would look like at the end of meiosis 1. But then if you go through meiosis 2 and these sister chromatids were broken apart, we would end up in number 1 here, we've got a full pink chromosome with lowercase b, lowercase d. That would be like this chromosome here, starting there and then going down to here. So start here, go down to here. That half would look like that. Then we've got uppercase B and lowercase d, and those would end up together. All right. So we've got 4 different ways in which those chromosomes can get broken apart there. In this case, we have the chromosomes that end up with lowercase d, lowercase b, or we end up with chromosomes that have uppercase B, uppercase D, and you can't find an exception to that rule amongst the 4 different possible outcomes for the gametes from those cells. However, when you look down here, because the crossover point happened above the point where the D genes were located, then we do end up with chromosomes in the end that are lowercase d, uppercase, or lowercase b, uppercase d. Sorry, I'm looking at it in the mirror image. On occasion, so there's 1, 2, 3, 4. So in all cases, each cell gamete that, or each cell that you ended up with after this sort of crossing over ended up with the different, with the chromosomes differently. So anyway, I think I botched the explanation there towards the end, but hopefully the main point is taken home, and that is these genes can act independently or dependently. The further apart they are from each other on the chromosomes, the more they act like independent genes on separate chromosomes, and the closer together, the less chance you have of crossing over happening between the genes. Okay, so in the next video, we're going to talk about that crossing over and making use of the percentage of times the traits are linked together versus not to map out genes, which is kind of getting to be maybe an obsolete variation of looking at genetics, because now we just go ahead and sequence the cell and figure out where all the genes are. But from a historical perspective and from an educational perspective, it's helpful to know that genes are sometimes on the same chromosome and sometimes on different chromosomes, and that might affect how those traits are going to be passed on to the next generation. All right. Genetic linkage is always tough to talk about, but I hope that video helped clear up any questions you might have had. All right, talk to you later. Like and subscribe or not.